Now that we've examined lipids and their contribution to membrane structure, let's now review the other major component of biological membranes, proteins. Proteins in a biological membrane may be associated with the bilayer in one of three ways. Peripheral proteins are loosely bound to the internal or external face of the membrane. They may bind by polar interactions, electrostatic interactions, or both, to either lipid or protein components. Note that these are non-covalent forces, so they can be separated from the membrane by relatively mild conditions, such as raising the ionic strength of the solution. An example shown here is a heterotrimeric G protein containing alpha, beta, and gamma subunits and associated with the membrane-bound protein rhodopsin. Rhodopsin itself represents another class of membrane-bound proteins, integral proteins. These are actually embedded in the membrane, that is, crossing through both layers of the lipid bilayer and having regions extended to both the external and internal membrane faces. These are so strongly embedded in the membrane that it takes more strenuous methods to isolate them, such as the use of strong detergents or bisonication. These vigorous methods may even lead to protein denaturation. Integral membrane proteins always exhibit secondary structure in the portion that crosses the bilayer. Recall that every protein contains polar amide bonds, and these would not react favorably with the very hydrophobic environment of the lipid interior. Remember that secondary structures form by hydrogen bonding interactions of the backbone amide bonds, and this serves to neutralize their polarity. These membrane segments may be one of the two main types of protein secondary structures that we've examined. They may be alpha helical segments, as pictured on the lower left, or they may be beta sheets, as illustrated on the right. Our final class of membrane proteins carries a lipid portion linked to a side chain. The lipid may be bound via a terminal amine group, as on the left, or via a cysteine residue, as in the center. Note that in each case, the lipid on the protein can readily associate with the lipids of the bilayer to anchor it to the membrane. If we compare these anchored proteins to the integral protein on the right, we can more readily see the distinction between them. Though each is bound to the membrane, the integral protein crosses entirely through the bilayer, whereas the anchored protein inserts its lipid component in only one of the monolayers. It may be anchored to either the inside or the outside of the membrane. In our next lesson, we'll take a look at a general overview of these membrane structures.